Okay, last but not the least, we have our final speaker for this afternoon, Dr. Sonja Schrepfer. So her research career has been dedicated to making fundamental discoveries in transplant and stem cell immunobiology. Dr. Schrepfer, professor at the University of California, San Francisco, and scientific founder and the senior vice president from Sonja Biotechnology, was trained as a cardiac surgeon in the cardiothoracic surgery departments in Munich and Hamburg, Germany, before pursuing a career as a full-time scientist. She received her PhD in transplant immunology and joined Stanford University to start her own research group in 2007 in the Department of Cardiothoracic Surgery. Dr. Schrepfer's findings have been highlighted in leading journals. Looking forward to your talk. I'll turn it over to you.
Great. Thank you very much for the kind introduction. So I would love to share data about overcoming the immunological hurdle in stem cell therapy and also how we started to identify. This is my disclosure slide. And as background for regenerative stem cell therapy, I wanted to share that my talk today is about iPSC, so the induced pluripotent stem cells. And one of the current concepts in the clinic is at the moment that we are thinking about autologous cell products. And that means that the somatic cell is been generated from a patient. The cells are then reprogrammed into the induced pluripotent stem cells and then differentiated into the respective cell product and transplanted into the very same patient. On the right side of the slide, you see another idea about allogenic HLA matched cell products, where basically iPSC banks are generated and they are transplanted into several patients which are HLA matched. And with this HLA matching, we try to overcome the immunological barrier because each cell does have like a fingerprint of the surface. We call that the HLA molecules, and that gets recognized by another person. And as soon as the immune system recognizes that as foreign, those cells or tissues will get rejected. What I wanted to share today is that in both approaches, on the left side, the so-called autologous cell product, so from the same patient, as well as when we create HLA-matched cell product banks, in both cases we have seen rejection. And the reason for that are antigens which are called MIHAs, the minor histocompatibility antigens. And I start with the HLA bank idea. The idea is that we match the HLA, so the fingerprint of the cells, from a donor to a recipient, but those two people are not related to each other. And for example, in the UK, the numbers would be that we would need 235 banked iPSCs to match the whole population of the UK. So that is basically feasible from a technical point of view. But I wanted to show you data that although we are creating HLA banked products, that those products have the risk of getting rejected. And for that, I want to show you this publication. It is from 2016, where the authors had used monkeys, rhesus macaque. And what they did is they generated iPSCs and then differentiated them into cardiomyocytes. And they had transplanted those cell products into HLA, or in that case, we call it MHC matched recipients. And what you see on the bottom is, in so-called group 4, when they perform those transplantations, 0% of the transplanted cells are surviving. When they now started immunosuppression of those monkeys in the middle, group 3, you see that again, 0% of those grafts are surviving. Although the immunosuppression, the tacrolimus that has been used, was dosed in quite high levels. So the 10 nanogram per ml trough level is something we are aiming for in a transplant patient. So that was not sufficient to prevent the rejection. When the authors then in group 1 used a very high and toxic immunosuppression regimen, using prednisolone, tacrolimus, and MMF, then you see those cells were able to survive. And these data show you that even if we think about HLA banking and matching this fingerprint of the cells to the patient, still we probably will see rejection and the patients will need immunosuppression. And the reason for that are the minor histocompatibility antigens, which we cannot match. Coming back to the autologous idea, here you see the iPSCs from the same patient gets transplanted back into the same patient, so that shouldn't cause any immune activation. However, because of the reprogramming, the differentiation, and the cell culture of those cells, there is the risk that those cells develop mutations in the mitochondrial DNA. And I show you data from our lab where you see that those mitochondrial DNA mutations are sufficient to cause a rejection, although the cell itself is from the same person, or in our experiment from the same mouse. So what we did here is we used the oocyte from that blue mouse. It was a black 6 mouse. We removed the nucleus and we put in the nucleus from the bulb C, the red mouse. So this cell product, these are pluripotent stem cells, should now be identical to the red mouse because all the information is coded by the nucleus. That's what we think. And if you transplant that back into the red mice, there shouldn't be a rejection. But the reality was different. If you look at that graph that shows you the growth curve of the transplanted cells, and you see in 6 mice, the cells are surviving, but in 4 mice, they got rejected. And the reason for that is that what you see here in blue in the oocyte, those mitochondria, they are having mitochondrial DNA. And this mitochondrial DNA can get recognized by the immune system and the cells get rejected. We performed mtDNA sequencing, and on the right side, you see that only 2 peptides, the COX-3 and the SUTB, were different in those mitochondria. And even those two differences, which are really minor, were sufficient to cause rejection. And you see in the immune analysis, this is an ELISpot assay, the high red bar. This is when we transplant the mitochondria from the blue mouse into the red. You see there is immune recognition. When we transplant the nucleus, which comes from the very same mouse, you don't see any immune recognition. So that really shows you that the mitochondria with only 2 differences can get seen and recognized and rejected by the immune system. So that is very important to keep in mind when we now think about the iPSCs. The iPSCs are induced pluripotent stem cells, and in theory, we could generate them for each patient. However, when we culture those cells, they have the risk to develop mtDNA mutations. And I show you that these mutations have the risk to get rejected, although the cells are from the very same patient. And that's what we did in this experiment. We used mice fibroblasts. We reprogrammed them to iPSCs. And then P7, that means that it's a very young iPSC generation. When we keep those cells now in culture, and around P37, we start to see changes in the mitochondrial DNA. And that's what you see in that heat map. The fibroblast, the fibrocyte here on the left, is basically the starter cell. Then we generated iPSCs. P7, it still looks okay, but at P37, you see that the COX-1 shows a mutation. And the heat map shows you the heteroplasmy, and that's 92%. That means that 92% of those mitochondria showed that mutation. And what happens is now, on the left side, when we transplant a P7 generation back into the same mice where we generated the cells from, the cells are surviving, no rejection. But you see on the right side, the blue graphs, when we transplant the P37 generation with the mutation, 3 out of the 5 mice rejected the transplant, although the cells were from the very same mouse. And that shows you that even autologous, so even generating iPSCs and going back into the same patient, is troublesome, and that we have to worry about rejection. And that brings me to the concept we are working on by creating hypoimmune cell products. And the underlying idea here is that we would have one healthy iPSC donor, very well characterized. We would receive cells, somatic cells from the donor, reprogram them to induce pluripotent stem cells, and then using gene editing to create hypoimmunogenic cells, which can be basically transplanted into any patient, but which don't get rejected because of the gene editing we do to those cells. we want to generate a universal cell product. And the advantages are that, of course, we wouldn't have immune rejection. And of course, we don't want to immunosuppress the patient. So it's really the transplantation of the cell and no medication. We then would be able to provide really good quality controls from that one off-the-shelf product. It's not so cumbersome to bank huge amounts of the cell lines because it's very well characterized and you can expand them. And it's much easier for manufacturing and quality control. And it's, of course, also a time issue. By the time a patient needs iPSC cell products, usually the patient is already sick. And that patient, of course, benefits from getting those cells immediately rather than having their own cells being generated and differentiated, which very often takes weeks to months. And there is no guarantee even that you can generate healthy cells from those patients. So that is the overall concept we are interested in. But the first step we had to do is understanding how those cells get rejected. And back then, if you look around 2005 was the time we started in that field, you find publication, they show you that pluripotent stem cells don't get rejected at all because they are so early in their development that they wouldn't be recognized by an immune system. So the first step for us was to understand, do they get rejected? And if so, which immune cell populations are responsible for that? So here we transplanted now pluripotent stem cells into different mice. On the left side, you see a mouse called bulb C. So these mice, they do have functional T cells, B cells, and K cells. When we transplant the cell product, you see that the pluripotent stem cells get rejected immediately. The curve hits the x-axis, the background, within 5 days. So that showed us those cells indeed get rejected. They are not immunoprivileged. We then moved on into the middle graph and we transplanted those cells into T-cell deficient mice. For example, about C nude mouse. Those mice do have functional B cells and NK cells, but they are lacking T cells. And what you see is that only 2 out of 9 mice rejected the cells. So that gives you a really good idea that the T cells are the major population responsible for the rejection of those cells. We then moved on to the right and we transplanted into mice with an perforin knockout. Those mice do have functional T cells and B cells, and only NK cells are not killing. But what you see, if you don't have NK cells, still all the cells can get rejected. So that really shows you that the T cells and the B cells are the major population responsible for the cells. So the idea we had was first, let's try to identify the molecules that are responsible for T cell recognition. And those are the molecules we already had discussed, the HLAs. That is basically the fingerprint on those cells. So the idea we had was, let's look into nature and see where we do have a so-called allograft that doesn't get rejected. And that happens, for example, in pregnancy. During pregnancy, 50% of the fetus have paternal proteins, so from the father. But the immune system of the mother doesn't reject the fetus. And it's not that the immune system is not able to see the fetus. The immune system is just not rejecting the fetus. I always compared like an army without weapons. So the immune system of the mother does recognize the fetus as allogenic, as foreign, but it doesn't reject it. There are 12 to 13 molecules responsible to achieve that. We call that feto-maternal tolerance. And the concept was we wanted to understand those molecules and find the fewest combination you need to achieve that hypoimmunogenicity. And at the same time, those molecules, of course, could not change the iPSC behavior or differentiation capacity, because the goal is that we want to generate those iPSCs and eventually differentiate them into cell types before we transplant that into patients. And I show you today the combination of the 3 molecules that seem to work in the models I show you today. So the first was wiping off the MHC class 1 molecule by removing the beta-2 microglobulin. And that is easy to achieve and really can get rid of the T cell and B cell response, because that is a fingerprint of the cells. And the number 2, the deletion of the MHC class 2 molecule, is exactly for the same reason, that we don't get a T cell response and B cell response. So those 2 molecules, if you remove them, the T cells and B cells are not recognizing the graft. But because as soon as you wipe off the fingerprint of those cells, other immune cells, innate immune cells like NK cells and macrophages, are sensing the missing fingerprint. We call that the missing self theory. And those innate immune cells can come in and kill immediately the transplanted cells. So we needed a molecule that prevented the cell transplant from getting killed by the innate immune cells after we had removed the fingerprint of the cells. And that was, in that case, the overexpression of the CD47. The idea then was to differentiate those hypoimmune iPSCs into different cell types. And really, with the underlying understanding, we want to provide off-the-shelf therapies to anyone, anywhere, anytime. And the mouse data here, we're using mouse iPSCs. And you see here the wild-type cells, so a normal mouse iPSC from a blue mouse transplanted into the same mouse, another blue mouse, formed teratoma. That's what you see on the left. So all the blue lines go up. So there was no rejection. But when we transplanted now the blue cells from a black 6 mouse into bulb C, into the red one, there was the recognition of the cells and rejection. You see that all the red lines, they stay flat. None of the cells survived. And we performed a battery of immune assays. You see here the examples for spots for the T cell response, and you see donor-specific antibodies for the B cell response. And what you see in blue is the background noise of the assay, so the transplantation of the same cell into the same mouse. And then you see in red the immune activation when we transplant into another mouse strain. The very same experiment is now shown on the right, but this time we used hypoimmune iPSCs. So the iPSCs where we did the gene editing, the MHC class 1 and class 2 knockout and the CD47 knock-in. And what you see on the bottom is that all the cells are surviving, all grafts are going up. And you see in the immune analysis that the red and the blue are at the same height. So the red transplant, the allogenic transplant, did not cause an immune activation in those mice. Because we don't want to transplant iPSCs into a patient, we need a cell product. And our lab was very interested in cardiac regeneration. We differentiated those cells into any cell type you find in the heart. And those are endothelial cells on the top. In the middle, you see the smooth muscle cells. On the bottom, the cardiomyocytes. And what that slide should show you, on the left side, we differentiated the mouse iPSCs wild type, on the right side the hypoimmune, and the differentiation worked the same. So the hypoimmune molecules we had introduced to the iPSCs did not affect the pluripotent state of the iPSCs nor the differentiation, which was very important for us. When we now transplant those differentiated cells into mice, you see on the left side, blue into blue, that's always the background control. Those endothelial cells are surviving. But if we transplant into the red mice, which is another mouse strain, you see in red the curves go down and those cells get rejected. The immune analysis on the bottom shows you similar data we have just reviewed for the iPSCs, but this time these are endothelial cells, and you see the red is much higher than the blue, showing the immune activation. On the right side, you see the hypoimmune cells, and you can see that the grafts stay up. The cells all survived for the follow-up here, in this case, was 50 days. And you see in the immune analysis that the red and the blue are at the comparable height. So there was no immune activation, even with those differentiated cells. We repeated the very same experiments with all these cell types, so the smooth muscle cells as well as the cardiomyocytes, with similar data. Here, that slide shows you smooth muscle cells. On the left side, the blue control stays up, the red ones get rejected. On the right side are smooth muscle cells which are hypoimmune, and you see the lines stay up, they are all surviving. The bottom, again, the immune analysis. On the left side, the red is much higher than the blue, immune activation. On the right side, they are at the same height. There was no recognition of the transplanted cells. And a similar picture you see on this slide for the cardiomyocytes. So we were able to demonstrate that not only on iPSCs, but all three differentiated cell types we were interested in. And what was really important for us to understand is getting rid of the T cell and B cell response is quite easy. All you have to do is wiping off that fingerprint, then you don't show the antigen to those cells. But as mentioned before, then you get the missing self theory and the innate cells. NK cells and macrophages are sensing that missing self and they kill immediately. So we spent a lot of effort identifying a molecule that could prevent the killing of those class 1, class 2 knockout cells. And these are killing assays I'm showing here. On the left side, these are normal iPSCs. When you incubate them with NK cells, natural killer cells, you see the blue line stays up. There is no killing. Normally, NK cells are not killing. But the next graph, which is the beta 2M knockout and the C2TA knockout, so this is the MHC class 1, class 2 knockout group. You see the NK cells are sensing the missing self and they kill the cells immediately. You see the blue line is crossing the x-axis. It's really fast, within 30 minutes. But if we now use the cells where we overexpress CD47, you see again that the line stays up. And that shows you that although we do have the missing self, the innate cells are not killing because the CD47 is protecting the cells. What you see on the right in the bottom is we block the CD47 molecule with an antibody. And if we block that signaling, then you see the cells get killed. And that really shows you the relevance of that molecule to prevent the cells from being killed by NK cells. We moved on with the research and into the human field. So here we generated exactly the same cells, but this time we generated human iPSCs. We then differentiated them into endothelial cells and cardiomyocytes again. And on that slide, we transplanted them into humanized mice. Humanized mice are mice which have been generated with a human immune system. And what you see on the left side, labeled with the endothelial cells Y type, is a human iPSC-derived endothelial cell transplanted into a humanized mouse. And you see that the signal declines within 21, 25 days. You see the curve goes down. When we repeat the very same transplantation and we are using hyperimmune iPSCs, differentiate them into endothelial cells and then transplant them, you see that the blue line is staying up. The follow-up here again was 50 days. And the cells did not get rejected. On the right side, you see the data for the cardiomyocytes. So similar result, the cardiomyocytes wild type get rejected, the blue lines go down. However, the hypoimmune iPSC-derived cardiomyocytes, the lines stay up. They did not get rejected. So what is novel about that platform is that we are thinking about preventing the T-cell B-cell response and preventing the innate immune response in separate ways. So first, the idea is to prevent the killing by NK cells. And that we do by wiping off that fingerprint. So I call that on that slide HLA A, B, C, E, G. That's basically the fingerprint of the cells. And we are removing the beta-2 microglobulin molecules, so what you see here in yellow. And by doing that, those molecules don't show peptides to the T cells. If they would show the peptides, you would see T cell activation. Now, colleagues in the field are now trying to inhibit the NK cells by overexpressing certain NK cells inhibitor. That could be HLA-C or E or G. And with those molecules, when you engineer them back in, you again now get peptide presentation, so the fingerprint is not completely wiped off. So what this platform here is doing, we are removing all HLAs, and then we control the macrophages and NK cells through a non-HLA dependent mechanism, and that is overexpression of the CD47 molecule. So in the last few minutes, I would love to show you some of our disease model data and really share with you our thoughts how we could help patients with the technology. So these are data here where we transplanted iPSCs derived endothelial cells, hypoimmune, into mice. with an hind limb ischemia model. And we then looked at different time points how those transplanted endothelial cells are doing. The cells we transplanted were queen positive, so luciferase positive. And then we used in red the VE Catarina marker to identify endothelial cells. And what you see around 2 to 3 weeks are those circular structures. That shows you that those endothelial cells start to form premature vessels. And if you now look after 5 to 6 weeks, you see those pinkish round structures in those tubes, and those are actually erythrocytes, red blood cells. So by looking at those data, we recognized that we could see some blood flow in those hypoimmune transplanted endothelial cells. And based on these data, we performed unhind limb ischemia by removing the blood flow of the mice. So these are perfusion Doppler images. And you see here on the right side, this red line that shows you the blood flow to the leg. On the other side, we had removed that artery completely. And then we had transplanted wild-type iPSC-derived endothelial cells, which get rejected, what I showed you. And what happens is that because there is no blood flow to that hind limb, the mouse is losing the limb. When we perform the very same model on day 1, you see here we had removed the whole blood flow to the leg. On day 7, you see some blood flow looks very chaotic, but if you now look at day 21 or 28, you see a beautiful blood flow to the leg, and those mice did not lose the limb. Although we had removed the artery, so the blood flow to the leg. So that showed us that hypoimmune cells can not only survive and don't get rejected, but they're actually functional as the cells we want them to be. And we had used an additional model for myocardial infarction just to look at a different disease. And what we did is we performed on myocardial infarction in mice and now transplanted iPSC-derived cardiomyocytes here on the left. And you see in bioluminescence imaging, the cells, as expected, they do get rejected. And you see here the data in blue, all the lines go down. When we transplanted hypoimmune iPSC-derived cardiomyocytes, you see the signal stays up in that myocardial infarction. That follow-up was 56 days. So the cells are all there in the heart. And when we look for the function of the heart, we could even see an improvement of cardiac function. We are looking here at PV loops, so pressure volume loops. On the x-axis, the volume, on the y-axis, the pressure. And in black, you see a healthy heart. So that is the volume and the pressure the heart is beating. In gray, you see when we now induce an infarct, the volume goes up because the heart cannot pump the volume out anymore. It's cardiac insufficiency. And then you see on the y-axis, the pressure goes down. That's what happens in a myocardial infarction. But on the right side, you see in red now, when we transplant hypoimmune cardiomyocytes, the volume goes down and the pressure goes up. So that shows you that these cells not only don't get rejected and survive, but they can really contribute to regeneration of a damaged organ. And that will not only, I think, change, of course, the options for patients, but that might also change the whole healthcare system. And of course, the life of a cardiac surgeon, for example. So I come from that area. Usually you perform your transplantations at 2 a.m. in the morning in solid organ transplantation. If we think now about regenerative medicine and cell transplants, it can be really 2 p.m. On a normal working day rather than in the night. So that might change the whole health care system and the hospitals. And I want to thank all the wonderful team members and the collaborators who put together all the data. Thank you.
Thank you so much, Dr. Sonja, for your great presentation and, of course, highlighting your experience in the industry. And I do want to open up the floor to some questions that the audience may have. I do see one question by Dr. Steve. He said, brilliant presentation, Dr. Sonja. iPSC-derived organoids may be of use for us looking at molecules involved in rejection. The 3D model plus immune cells plus confocal for live cell imaging of cell killing is possible. If of interest, please contact me, Dr. Miller.
I will. Thank you. Thank you.
Well,
Great.
Hi, Sonja, Devanu here. Great talk. Thanks a lot. I work on the crystallography and structural biology of some of the pluripotent transcription factors, SOX2, NANOG, and OCT4, and in fact, worked on the crystal structure of NANOG protein DNA complex with Bruce Conklin and Shinya Yamanaka. And it's so very nice to see that the ability to use protein factors to do induced pluripotent stem cells, the effort is ongoing and continues to grow. So it's very nice to see that.
Thank you.
Any other questions for Dr. Sonja in the room? We can take a couple more. And I do see, Dr. Sonja, we do have quite a few people from your group in the audience from Seno Biotech as well. That's great.
Nice. Great to see you all, guys.
Yeah, absolutely. And yeah, Dr. Sonja, if you do have a moment, do please tune in for the panel discussion after your talk. I believe it's about emerging strategies for ensuring success from drug discovery to preclinical research. So if you have a moment, please.
Thank you. It was a pleasure being here today with you. Thank you.
Absolute pleasure having you. Thank you again.